Hello, hello. My name is Raphael Balogun. Today we're talking about the notable fungal biochemistry, and the notable fungal biochemistry we're talking about right now is their mode of nutrition, how they do their biochemical reaction to exchange nutrients, mode of nutrition. We're going to be talking about their mode of excretion. We're also going to be talking about their byproduct, that is the mode of excretion. We're talking about the fungal and resistance principles as well, how they actually have antifungal and resistance principles, how they get resistance to antibiotics and the rest. You understand what I'm saying? Now, starting from the first one, which is the mode of nutrition, there are three major modes of nutrition in mycology, in fungi. The first one is, since these organisms are naturally heterotrophic in nature and they cannot digest internally, these organisms can only digest externally. That's one of the differences between a fungi and an animal. They digest externally. Fungi cannot digest internally. Fungi can only digest externally. So they secrete whatever they want to secrete to digest it, and then they now absorb. So in this way, there are three major ways at which they digest and absorb. Now, there is the first mode of nutrition, which is the most primordial and belongs to this category, the direct active absorption. Now, in the direct active absorption, this fungi does not degrade sugar to anything again, does not do anything than to consume the sugar directly. You get what I'm saying now? It doesn't break it down to any other thing. It consumes the sugar, it consumes the nutrients directly from the environment. That is why this kind of organisms will only be in a moist place. They cannot really survive. They will survive, but they will not grow. They will not grow and reproduce in a dry condition. These are the only fungi that requires moist to grow. Yeast and any other ones like the Candida species and the different types of other species that are unicellular in nature. So every unicellular fungi are the ones that use the direct active absorption, which they use active transport mechanism to absorb the nutrients they need into their body from the environment. That is directly. They don't digest, they don't secrete anything than to keep absorbing. They keep absorbing and absorbing. So when they now absorb, they now excrete their byproduct. Now their byproduct in different ways. For the case of yeast, is carbon dioxide. For the case of amoeba, candida, whatever is anything that now causes that irritation in your vagina, because they don't actually. And also that is one. And there are also some species who, yes, still do some temporary kind of digestion, which is trick a little bit, but most of it is through active means. That is the first one. Then we'll talk about the second level, which is the direct enzymic absorption. Now, the direct enzymic absorption is this category of it can be mold, it can be anything that can actually eat anything that is still drier than water. So it can be any wood that has some moist on it, just a little moist, or any other stuff like that. It may be dry too sharp, but it should be able to contain a little amount of water. So in this case, this organism secrete enzymes on top of that organism it wants to degrade, and then once it secrete that enzyme on it, then once the enzyme is secreted, the organism, the food digests to the one it can absorb, sugar and any other thing it needs, which it consumes. And once it consumes it, then that is how it is. It has already digested. That is another mode of digestion. While the maximum and highest one is the hyper penetration absorption method. This hyper penetration method is the filamentous and the mushroom-like one, which they actually start to input their roots. I call it Raphael mycelium. They start to put it and use it to penetrate things like wood, things like hard soil, things like soil, things like dogs. You get what I'm saying? They start to penetrate. They penetrate, and once they penetrate everything, they start to absorb. And once they penetrate it, they now release the enzymes. Once they release the enzyme, that one digests the one in their immediate, and then it falls back on the mycelium, the hyphae, and then it's actually absorbed to the whole cell or cellular colony. That is the last one, but the biggest and the most complicated one. That is it about the mode of nutrition, and a lot of biochemistry happens during the process. The enzyme itself actually degrading is a biochemistry in nature, and this enzyme for digestion is what most scientists actually extract as what? As antibacterials, you get, or antibiotics. You understand what I'm saying now? But there's a problem with this, all these antibiotics. If a chemical can digest a bacteria, be careful, it can also digest a human cell or a living tissue. You understand what I'm saying? Especially when the living tissue has a kind of weak membrane. You understand what I'm saying? So that is that about that. So we are also going to think about their byproduct classification. Now, there are different fungi with different types of byproducts. These byproducts have been used to name them many times. For instance, penicillin and which secrete penicillium, which secretes penicillin. Penicillin is actually two things. One, the byproduct of the food it has eaten to produce the enzyme penicillin, and at the same time an enzyme to get more intake. So always keep in mind, in the case of fungi or anything you get that secrete things, or get anything that secretes things to get more, you get what I'm saying? The byproduct is actually that thing is secret, is one of the byproducts. Then there will be other byproducts like carbon dioxide and water and some other degraded materials like that, plus the enzyme that is also secreted. Everything are the byproducts of that fungi. So the byproduct of fungi, you can classify bacteria and fungi based on that, and it's also a biochemistry. The biochemistry of developing that penicillin is one. The biochemistry of and then the food and the action of penicillin on other organisms and the environment and other foods that it consumes is another. You get, and there are other types of enzymes that is different from penicillin or streptomycin, produced by different types of fungi to degrade food and also to digest bacteria and all those stuff. So that is that about the byproduct of classification. Then there's the antifungal and resistance principle. Now, keep in mind, even though they also produce antibiotics that we use to fight bacteria and all those stuffs, they produce it to eat, not to fight them. They produce them to eat them, and then we to take them to do it. Now, note, all these organisms you get also are parasitizing human beings, and then they have a way of escaping us from using drugs. So sometimes you use drug and you don't work on them because they gain resistance. The way of resistance is similar to that of bacteria, because resistance happens on a cellular level, not on an organismal level. Like any organismal level of change you get that is actually biochemical in nature. When it comes to chemistry and its chemistry, then note it's always cellular in nature, because don't forget, cells are made up of chemistry and physics. So you must always keep that in mind. Laws of physics bind cells together and keep them working together. Law of physics binds molecules together and make them create the long chain and whatever that makes you see the matter. Law of physics keep the activity going and keep them alive. The biology is now the one that's combines both of them from the life perspective to see the mechanism between both of them. Just like you can use physics and chemistry to develop a car, which is a metal living organism designed by mankind. That is how physics and chemistry was also used to create human beings, which is designed by God, our creator. So that's how that happens. So there are different principles at which they resist all these things. Just like bacteria, once you introduce an antibiotics, you get selection of the fittest first happens. Most of them will die. The ones that are strong enough, or the ones that are luckiest enough, let's say selection of the fittest and luckiest, those that are fittest enough and those that are lucky enough to escape at that point in time would have the memory of the chemicals used against them, or the substance used against them. And I have told you how that memory works. It's not like they just store the codes or whatever. They have a copy, a sample of that toxin in them somewhere. Even if it is damaging them, they keep it within their body and start to learn how to neutralize it. So whatever that has successfully neutralized it will be recoded and encoded back to their genome, which they will transfer during sexual or asexual reproduction, and then they build a new colony or a new civilization of organisms that do what? That is resistant to that of antifungal or antibiotics. So that is all you need to know right now. Take care and invite more people to take these courses. Thank you.